I think I might not have paid for my Screencast-O-Matic subscription, or maybe I don't know, something's happened. I can only do 15 minutes at a time. I will fix that problem in a bit. So what I was just about to say is just that gherkin is a ligand. So the torpedo receptor is an epidermal growth factor receptor, and it initiates one of the kinase cascades. So if you go back to lecture 4, this is one of those kind of relays where the phosphorylation, subsequent phosphorylation of a bunch of different molecules is required until finally the one of the kinases goes inside the nucleus and phosphorylates, activates transcription factor. So that's the kind of signaling that this is. And gherkin is the ligand, and the receptor is called torpedo. Torpedo is one of the first things I worked on when I came back to science after a short break doing other jobs for a while. So it's very near and dear to my heart. Okay, so the gherkin then it's a signal, and the torpedo receptor is in all the follicle cells surrounding, but only the ones on the dorsal get the actual signal, the ligand binding. And so you get, and this is just an example of one of the proteins that gets expressed in response to gherkin signaling. And you can see, hopefully, that there's a ton of it on the dorsal side. And because the egg is actually round, so we're only seeing like a slice through, but this shadow is also showing that, like, as you come around from the most dorsal, there's still some protein, and it doesn't really fade away until it's sort of around here, and then you don't have very much on the ventral side. So this is a dorsal versus ventral gradient. And so, and the response really for this particular signal is there are a dorsal identity is adopted here, and basically certain proteins are produced that are going to be required for developing those dorsal appendages and some of the other proteins and so on that are specific to the dorsal side. But there is also a kind of a repression. The message is, okay, don't make any of the pipe protein. The name of the protein is pipe. And so these guys, they don't get that signal, and so they are ventral as a kind of default. They don't receive the gherkin signal, so they go ahead and they make the pipe protein. And this is part of the ventral specification in the embryo. And I will just quickly remind you about toll signaling, and then I will add an extra piece of information about what the pipe protein is doing in this process. Okay, so you remember that all the way around the egg membrane, you have the toll receptor, and the spatula fragment, the ligand part of spatula, is only produced in the ventral region, and so therefore we only have really strong toll signaling on the ventral side. And the signaling leads to, and you can review for yourself those proteins involved, but leads to the degradation of cactus and then the nuclear localization of the dorsal protein. And so on the ventral side, you have lots of dorsal protein in the nucleus, not so much on the sides, and none up top. So that up top, you have your DPP signaling, and then on the bottom here, you have ventral specific, mesoderm specific proteins being produced, like snail and twist, and rhomboid and SOG, and so on the side. So a bunch of those. So how is this connected to pipe? It turns out that pipe is part of a whole cascade of proteins, and I only want to mention a couple of them, but a whole cascade of proteins that are required to go from an intact spatula protein to just the production of the cleaved form and the little fragment of it that acts as a ligand. So you have spatula, the whole full protein, all around the perivitelline space, this little space between the outer membrane and the vitellin membrane, but it's only cleaved on the ventral side. And so this is a picture from this site, and it basically just shows that on the in the ventral follicle cells, the pipe protein is being produced, and on the membrane, you have a target protein for pipe, which we don't even we don't even name. It's just it's called pipe target. Um, you know, these proteins are found in very tiny amounts, and it's extremely difficult to study them because they're present in this teeny weeny little part of the egg in a very localized way. So you need buckets of the embryos in order to isolate everything. It's technically really very difficult. But this pipe target protein, oops, um, basically takes the Easter protein, which is the one I had introduced to you, and it cleaves it in order that it then can cleave spatula and make the toll ligand. So there's a series, and there's another protein called snake, and there's other things going on, but this is what you need to know. There's a pipe protein. Sorry, a pipe target. It's a target of pipe. So pipe modifies it in some way. That modification causes it to cut. The Easter protein, part of it is degraded and part of it is activated by being cleaved. It then cleaves the spatula into the cleaved form, which is the active ligand. Yeah, and it's just, there's a whole lot of steps in between here that we don't know very much about, and so I'd rather you just learn a couple of steps sort of at this level, and we can leave it at that. Okay, so this is a summary of the important molecules that you need to remember. The bicoid messenger RNA ends up at the anterior end, and you need to know the communication between the oocytes and the surrounding cells, and how one egg chamber is sending information to the one ahead of it about anterior versus posterior. And then the nanos RNA via OSCAR, a protein called OSCAR, which is the one that kind of localizes nanos to the posterior. And just notice they're being produced in the nurse cells, and then they're being transmitted via microtubules into the egg itself and into the correct locations. Those are going to be translated after fertilization to make the protein gradients. Torso-like, we haven't really talked very much, but torso-like is a specific probable ligand for the torso receptor, and it's just deposited in the very most polar anterior and posterior cells, and that's all you need to know about it, because we didn't really talk much about that. And then on the ventral side, as a result of Gherkin signaling, on the dorsal side, pipe is laid down just in the ventralmost cells, which leads to the production of the spatula, not the protein, but the ligand, right? The ligand part, the cleaved form of it. Okay, so that's a summary of the basics of what happens. In addition to, you know, how the egg is formed and so on. That stuff is really basic as well. And I just draw your attention at the end of the sections of the textbook. You've probably seen these already, but this basically is worth looking at, these, because these really summarize the basic steps and the most important molecules involved.